Welcome to The Explainer. Today we're going microscopic. We're embarking on a journey right into the heart of the living cell. We're going to explore the essential toolkit that scientists use to unlock its mysteries. Everything from how they keep cells alive outside the body to how they create these stunningly detailed maps of their internal machinery. This, right here, is where modern biology truly begins.
So we're basically going to follow the exact workflow a researcher uses. We'll start with how to grow cells in the first place, then we'll look at how to gently crack them open, sort out all their internal parts, and finally actually see what's in there. And we'll wrap it all up by connecting these techniques to real-world, life-saving medicines.
Okay, so first things first. Before scientists can even think about studying cells, they have to grow them. And our journey here begins with a story that's not just foundational, but also incredibly complex from an ethical standpoint. A story that really changed medicine forever. Back in 1951, cells were taken from a woman named Henrietta Lacks, completely without her consent. These cells, later named HeLa, turned out to be the first human cells ever to grow indefinitely in a lab. The HeLa line became the absolute cornerstone of modern cell biology. It's been used for everything from the polio vaccine to cancer research. And it really forces us to think about the profound impact and the huge ethical questions that surround this kind of work.
So what is cell culture? Well, at its core, you can think of it as creating a five-star hotel for cells right in the lab. Scientists have to provide absolutely everything a cell needs not just to survive, but to actually thrive, completely separate from the organism it came from.
And to keep these cells happy, you have to be a meticulous architect of their little world. They need a perfect nutrient soup, the right chemical signals from serum to tell them to grow, a constant, cozy body temperature, and a perfectly balanced pH. And all of this has to happen while you're waging a constant war against bacteria and other microbes that would love to contaminate everything.
All right, so let's say we've succeeded. We now have billions of healthy, growing cells. You'd think the hard part is over, but our work has really just begun. The next big challenge is getting inside them to study their parts, and the real trick is to do it without smashing everything to bits. Think of this process, homogenization, as a sort of controlled break-in of the cell. The goal isn't destruction. It's about carefully cracking open that outer wall just enough to let all the precious content spill out totally intact and ready for us to analyze. Now, researchers have a whole toolkit for this break-in. They can use tiny grinding beads, they can blast the cells with high-frequency sound waves, or they can even just dissolve the membrane with special detergents. And the choice of tool is absolutely critical. For instance, a gentle osmotic shock works great on animal cells, but it would be completely useless against the tough, rigid walls of a plant cell.
And this really gets to the heart of the challenge. It's the difference between using a scalpel and a sledgehammer. If you use too much force, all those delicate little organelles you want to study, like the mitochondria or the nucleus, they're just instantly destroyed. The success of the entire experiment really hinges on getting this one crucial step right.
So, what we have now is basically a microscopic soup of all the cell's parts, a chaotic mix of every single organelle. So how in the world do you sort through all that? Well, the answer is surprisingly simple. You spin it. And you spin it really, really fast.
What we use is a centrifuge, which is basically a high-speed spinner that generates incredible force. Think of it like a salad spinner on steroids. This force separates all the cell's components by their weight and their density. It forces the heaviest parts down to the bottom first, kind of like how rocks would sink way faster than sand in water.
Scientists have two main strategies for this. Differential centrifugation is like a rough sort, using a series of sieves to separate big rocks from smaller pebbles. But for a much more precise separation, they use something called a density gradient. This creates layers of liquid, and each organelle just floats to the one specific layer that perfectly matches its own density. It gives you these beautifully clean, pure fractions.
This step-by-step process is just brilliant in its simplicity. With each spin, you just crank up the speed. The first, slowest spin gets the heaviest stuff, like the nucleus, to form a pellet at the bottom. Then you take the leftover liquid, pour it into a new tube, and spin it faster to get the next heaviest things, like mitochondria. And you just keep doing that, over and over, until only the tiniest molecules are left floating around. But how does scientists actually know their separation worked? How do they know their samples are pure? Well, they run a purity check using what are called marker enzymes. These are unique proteins that act like a specific ID tag for each organelle. So if the mitochondrial enzyme suddenly shows up in your nucleus sample, you know you've got contamination. It's the ultimate biochemical quality control.
Okay, so we've isolated all the cell's components, we've separated them into these neat little test tubes, but how do we actually see these incredibly small structures? Well, that's where the microscope comes in. But as you will see, not just any microscope will do.
You see, to truly look inside a cell, you need two things. Magnification is the easy part. That just makes the image bigger. But the real key, the holy grail, is resolution. That's the ability to see fine detail with absolute clarity. You can magnify a blurry image as much as you want, but without good resolution, it's pretty much useless.
Now, a standard bright field microscope is a fantastic starting point, but it's got some major limitations. Because the wavelength of light itself is relatively large, the finest, most intricate details of the cell just stay hidden. It's sort of like trying to build a tiny, delicate model ship while you're wearing big, clumsy mittens. You just can't get to the detail.
So, to see beyond the limits of light, scientists had to come up with a completely new approach. They developed this incredibly ingenious technique called freeze etching. The core idea is to bypass light completely. Instead, you create an ultra-detailed metal replica of the cell's interior, a perfect little cast that can then be viewed with a much more powerful electron microscope.
This whole multi-step process is kind of like cellular archaeology. First, the cell is flash frozen. Then it's cracked open with a tiny blade. Then a thin layer of ice is evaporated away, which exposes the surfaces of all the organelles. This newly revealed landscape is then sprayed with a micro-thin layer of platinum and carbon, which creates a perfect metal cast of everything.
And the result is, wow, it's just breathtaking. We're no longer looking at fuzzy blobs of light. Thanks to this technique, we can see the individual proteins studded across the cell membrane. We are literally looking at the machinery of life in stunning, three-dimensional detail.
So, let's recap. We've grown the cells, we've broken them open, we've sorted their parts, and we've visualized them in incredible detail. But what's the point? Why does all this painstaking, complicated lab work actually matter? Well, the answer is what changes modern medicine.
You see, every single technique we've just talked about can be scaled up. That ability to grow cells in a tiny little dish can be expanded to grow them in massive industrial vats. And when you do that, you turn these cells into tiny factories for producing the powerful, life-saving medicines we call biologics.
I mean, just look at this list. EPO for anemia, factor VIII for hemophilia, TPA, which dissolves blood clots after a stroke, and monoclonal antibodies to fight cancer. Every single one of these is a complex protein that's produced in massive, large-scale animal cell cultures, all using the very same methods of growing, purifying, and analyzing that we've just explored.
So by learning how to take the cell apart, piece by piece, we've managed to transform it into this powerful factory for modern medicine. We've developed treatments for diseases that were once considered completely untreatable. And this whole journey inside the cell leaves us with one really thrilling question. What other secrets is it waiting to reveal? What new cures are just waiting for us to find them?